Imagine two people watching a video. What are the chances they're watching the same video? Not very likely, right? Now, consider this: what are the odds that two people would need identical medical care throughout their lives? Those chances are even slimmer. As we age, each of us develops different health needs. To address this, researchers and doctors are constantly working on ways to tailor medical treatments. One promising approach involves using stem cells. So, what exactly are stem cells? They're special because they haven't yet been assigned a specific role in the body. While skin cells protect, muscle cells contract, and nerve cells transmit signals, stem cells don't have a defined function. However, they have the potential to transform into any other type of cell your body needs. Your body uses stem cells to replace old, worn-out cells. Scientists hope that stem cells can one day lead to personalized medicine, where we could regenerate damaged or aging body parts with new, healthy tissue. This idea, known as regenerative medicine, involves using stem cells to create tissues or organs that can replace those affected by injury or disease. There are different kinds of stem cells being studied for medical use. Adult stem cells, or tissue-specific stem cells, are found in small amounts throughout your body. They help maintain and repair tissues by replacing cells that have naturally worn out. Embryonic stem cells, however, come from donated embryos from fertility treatments. Unlike tissue-specific stem cells, these are pluripotent, meaning they can develop into any type of cell in the body. There's also a third type of stem cell called induced pluripotent stem cells. These are regular cells like skin or liver cells that scientists have modified to act like embryonic stem cells, giving them the ability to turn into any cell type. While the main goal is to use these stem cells to grow new tissues and heal the body, scientists also use them to study how the body works. But there are ethical concerns regarding the research of stem cells. One of the major ethical issues in stem cell research centers on the use of embryonic stem cells, which are derived from early-stage human embryos. These cells, obtained from embryos typically donated from fertility clinics. Obtaining these cells usually involves the destruction of the embryo, which sparks a range of ethical concerns, particularly around the moral status of embryos. Many people believe that human life begins at conception, which, to them, implies that even early-stage embryos have a right to life. This perspective is often grounded in religious beliefs, where the sanctity of life is a core value, leading opponents to argue that embryonic stem cell research crosses a moral boundary by sacrificing potential life for medical advancements. On the other hand, proponents of embryonic stem cell research argue that these cells hold tremendous potential to cure or alleviate the suffering caused by diseases such as Parkinson's, Alzheimer's, diabetes, and various forms of cancer. From this perspective, the potential to relieve human suffering outweighs the ethical concerns surrounding embryo destruction, especially when embryos are donated voluntarily and would otherwise be discarded by fertility clinics. Supporters also point to the fact that embryonic stem cell research has paved the way for significant scientific breakthroughs, arguing that halting research could hinder our ability to develop new treatments that could benefit millions. Induced pluripotent stem cells, iPSCs, offer a partial solution to these ethical dilemmas. These cells are adult cells, like skin or liver cells, that scientists have reprogrammed to function like embryonic stem cells. iPSCs can differentiate into a wide range of cell types without requiring embryonic sources, addressing some moral concerns. However, iPSCs are not yet as reliable or efficient as embryonic stem cells in many therapeutic applications. There are also safety concerns with iPSCs, as they sometimes develop genetic abnormalities during reprogramming, making them risky for certain treatments. This has led to ongoing debate about whether iPSCs can fully replace embryonic stem cells in research, leaving embryonic cells still essential for some studies. Animal testing in stem cell research is another significant ethical issue. Before moving to human trials, scientists test stem cell therapies on animals to assess safety and efficacy. Animal models, especially mice, are used to predict how treatments might work in humans, but these procedures can lead to pain, distress, or even death for the animals involved. Critics argue that animal testing is ethically problematic, particularly when animals suffer without clear benefits or when alternative methods might exist. There is also concern over the welfare and humane treatment of lab animals, as critics argue that we should strive to minimize or eliminate their use wherever possible. Balancing the need for preclinical testing to ensure human safety with animal welfare concerns is a challenging aspect of stem cell research ethics. In summary, the ethical issues in stem cell research are multi-layered, involving deeply held beliefs about the moral status of human embryos, the treatment and welfare of animals, and the potential for stem cells to treat devastating illnesses. Balancing these concerns means weighing the promise of scientific advancement against the respect for life and welfare. Many scientists and ethicists argue that ongoing discussion and stringent regulations are necessary to ensure that stem cell research is conducted responsibly, ethically, and with the public's trust.